everyone. Uh, in the previous video we have seen about the human reproduction NCERT Exemplar MCQs. Now today let us discuss about the biotechnology principle and process chapter, some questions from the NCERT Exemplar, some PYQ we will see after this, but first of all we will solve the NCERT Exemplar, then we will go to solve the other MCQs or the previous year question, which is very important. Okay, first we will see the NCERT Exemplar of this chapter. Okay, we will see the NCERT example. Okay, so let us continue with a good spirit. Let us see biotechnology and its principle, right? So first of all, what they are asking in the question, that rising of the dough is due to what? So rising of the dough means it is actually the process of the fermentation, right? It is the process of the fermentation. So in the fermentation we have seen the dough has been raised up. Because of that, what was the result of the fermentation? Because of that the rising of the dough was happening. It was actually high CO2 concentration. CO2 was increasing. So because of that the production of the CO2 was increasing, right? Next question is, which of the following enzymes catalyze the removal of the nucleotide from the ends of the DNA? From the ends means they are actually asking from the restriction enzyme. It is from the restriction enzyme. In the restriction enzyme we have seen two types, that is the exonuclease and the endonuclease. Exonuclease and endonuclease. Exo means from the ends and endo means from the middle. From the middle, right? And exo means from end. Okay, so here they are asking in the question from the ends, so we will go with the exonuclease, right? So second question is completed, right? Now the third question is, they are saying the transfer of the genetic material from one bacterium to the another through the mediation of the viral vector is termed as what? So this is actually the mode of the transformation of genetic material from one cell to the another cell. So there are actually the three types. This is the first one is a conjugation. Second would be the transformation, and third would be the transduction. Third would be transduction. Okay, so in the conjugation what was happening, from one cell to the another cell there was actually the tube will be forming, one tube will be forming in between the two cells, like this. This is another cell, so this tube is known as a conjugation tube. This is known as the conjugation tube. So on the basis of that we will call this process that genetic material will roll from here and will enter into the another cell. So this is the conjugation process. In the transformation from one cell to the another cell, actually there would be a degradation of the one cell, okay, and because of that the membrane will be disrupting. Membrane would be disrupting, and the another cell, the genetic material will come out and it will be taken up by the another cell. So this is the transformation process. And the third one is very important, which is asked into the question, that from one cell to the another cell how the transformation is occurring. This is the one cell, this is the another cell. On the one cell the bacteriophage or the like any viral vector will set on it, will take the genetic material and will go to the another cell and transform it to the another cell, like this. So this process is known as a transduction. So here bacteriophage is actually using bacteriophages or either any virus, so viral transformation we will call it. Okay, so this is what asked into the question, so we will go with, because they are asking about the viral, so we will go with the transduction process. And remaining two, I hope you would be clear with these two all. And translation you knows very well, the protein synthesis, that is a completely different process. It is a protein synthesis. Okay, now let's see the next question is, which of the given statement is correct in the context of the visualized DNA molecule separated by the agarose gel electrophoresis? In the agarose gel electrophoresis, the DNA fragment or the DNA molecule which is separated, how we can see it? By using the ethidium bromide on the stained DNA stained with the ethidium bromide, ETBR. Okay, and then we will use the UV light to see, to visualize it. Okay, so this would be the answer, ethidium bromide, but visible light cannot be seen in any visible light, like whatever we are using in our homes, that is not visible into that light. Under the UV light only it would be visible. The next question is here. Let us see the next question. They are asking in it that restriction in the restriction enzyme refers to what? Restriction in the restriction enzyme means, what is the meaning of the restriction word in the restriction enzyme? So restriction means to prevent. Okay, restriction means to prevent, and restriction enzyme means the function of the enzyme is to cut. Okay, so to cutting they are not asking, they are asking what is the meaning of the restriction in this meaning, in this particular word. So that is actually to prevent the multiplication of bacteriophage by the host bacteria. Okay, it is not the cutting. Cutting is actually the function. Cutting and cleaving is actually the function of the restriction enzyme. They are not asking that. They are asking only the meaning of the restriction. So we will go with the third one. Next question is question number 6. That is, which of the following is not required in the preparation of the recombinant DNA molecule? What is not required? Means we need a DNA molecule. In this we want to add our genome, and then we want to make it as a recombinant DNA. If like this, so our genetic material should be inserted into it. We want to make it as a recombinant. I did not make it round because it is not a plasmid, so it is a recombinant DNA, right? So what is not required, they are asking. Restriction endonuclease, it is, if we will cut, then only we can insert our gene of interest. Then for ligation, we will use a DNA ligase to ligate with the help of the restriction. With the help of the DNA ligase, we will stick it, or we will keep this gene of interest into the host cell. Okay, and the fragment of DNA is actually the gene of interest our. But E. coli is the part of the transformation. After making it, we will transform it. Okay, so it is a part of the transformation step. Okay, it will be the part of the transformation step, but they are not asking in the question about the transformation step of complete RDT. Okay, rDNA technology. In that, transformation was a step in which E. coli or the bacteria cell we was using, but they are asking only to make the first step, to complete the first step. Okay, so because of that we will need all these, but not transforming into the cell. They are asking from the first step. We will go with the fourth one. Okay, this is not required. Next is, in a agarose gel electrophoresis, DNA molecule are separated on the basis of their what? Actually, in the gel electrophoresis, we have taken one agarose gel plate. We have taken our agarose gel plate, and in that we have loaded our DNA fragment, right? And then after, based upon the charge and based upon the size, if the smaller size is there of the DNA, so it would be moving very fastly. Smaller and the light weighted would be very fastly go to the opposite end. Okay, here is the negative end, there is a positive end. Okay, it is an anode, here would be the cathode. DNA is in itself is a negative charge. DNA is negative charge. So we can say DNA is negative charge, it would be try to move very fastly towards the positive end, towards the positive pole. Okay, so it is depend upon the charge as well as the size also. Okay, and what is the ratio between them, that also, charge should be high. Okay, as well as the size also should be very low. Small size, small fragment will move very fastly. It is just like if we are running in a race, those person who are having very less weight, low weight, they will fastly run in the race and they will win. The person who are very healthy, fatty, so that person will not able to run very fastly. Okay, just like that only. So it is depend upon the charge, size as well as the charge to the size ratio. So we will go with the all. Okay. Then next question is, the most important feature of the plasmid to serve as a vector in a cloning experiment. What is important? It is not given. I think it would be there in the NCERT lines that ORI is there. ORI is actually the controlling the copy number. Control copy number. Okay, so that is what they are asking, that what is the main feature. These all are the features. These all are the features of the plasmid. But what is the most important among them? That is the origin of copy of the replication, which is actually the control of the copy number. If it is not controlling, so whether it would be very good plasmid, but ORI is not good, so complete process would be ruined, right? So we will go with the first one. Question number 9 is, while isolating the DNA from the bacteria, which of the following enzyme is not required? So for that we should know that we are having a test tube, for example, and in that test tube we are having one cell. In this test tube we are having a cell, and we know in a cell we are having everything. We are having the nucleus, ovaries, endoplasmic reticulum is there, mitochondria would be there, proteins, RNA, DNA, everything would be there. So if we are having this cell into this test tube and in the distilled water or something, some solution in which the cell can survive, so how to reach up to the DNA? Because we need our DNA from this test tube. What we need our DNA from this test tube. Okay, and in the nucleus, I show you outside. This is the cell, this is the nucleus, this is the endoplasmic reticulum. Okay, this is the chromosome and the DNA. Okay, here would be the RNA also. Here are some proteins, here are the mitochondria, here would be lysosomes, Golgi bodies, everything would be there. Okay, so to reach up to the DNA, what we should degrade first? First we have to degrade the cell membrane, then any protein or anything, any enzyme, any hormone, any molecule, any ion which will come in pathway, that all we have to degrade. We have to degrade the endoplasmic reticulum membrane, we have to degrade the nucleus membrane, inner membrane of the nucleus, which is actually attached with the DNA fragments, DNA molecule or the chromosomes. Then only we will able to reach to the DNA. So what we have to degrade? We have to degrade the membrane, which is made up of the lipid. We have to degrade the protein, which may be any ion, which may be any other kind, any kind of the protein, which can be any enzyme, which can be any hormone. Okay, then after we have to degrade the RNA if it will come into the pathway. Okay, then we will reach finally to the DNA. So to degrade the lipid, we will use the lipase enzyme. We will use the lipases. To degrade the protein, we will be needed proteases. Proteases. To degrade the RNA, we will use the RNases or RNAs. To degrade the DNA, we use the DNAs, but we actually require the DNA. So we will use all these three, but we will not use the DNAs. If we will use the DNAs, our DNA will degrade, but we need this DNA. Okay, so in the question as they are asking, while isolating the DNA from the bacteria, which of the following enzyme is not required? Okay, which is not required. So based upon that technique only we can solve the questions here, that if we need to isolate the DNA from the bacteria, so obviously we can call there would be the deoxyribonuclease. It is what? Deoxyribonuclease is what? DNase, right? Protease would be there, lysozymes would be there, ribonucleases would be there. So the all we should degrade, but we don't want to degrade the DNase, deoxyribonuclease. Okay, so this is the full form of the DNase, is the deoxyribonuclease. So we will not use this. This is not required. Remaining to degrade the nucleus, RNA, ribonucleases we will use. For lysozymes, protease means to degrade the protein. Okay, lysozyme is what? It is a kind of the enzyme, and enzyme is what? It is a protein in nature. So we will use the proteins. Here we are using, here also it can be degraded by that. So we will not use the DNAs. Okay, I hope this question will be clear with this experiment. Now next question number 10 is, which of the following contributed in popularizing the polymerase chain reaction technique? Means among these four, which is of the following will contributed more in popularizing the PCR technique? Okay, so actually this is all depend, PCR technique is actually in which the temperature would become high and the low. It is all depend upon the temperature. If the temperature is high and the temperature is low, so based upon that we have isolated a enzyme Taq polymerase. In place of the DNA polymerase we are using the Taq polymerase because this is isolated from the Thermus aquaticus. Okay, which is a bacteria which can survive at a very high temperature, even at the 80 or 90 degrees Celsius. Our body enzyme cannot survive at that temperature. Even at the 40 degrees Celsius the enzyme will become degrade, but the Taq polymerase can survive. So it is all contributed the PCR technique, the thermostability of the DNA polymerase. Which DNA polymerase? Actually we are calling the DNA polymerase the Taq polymerase based upon the function of it. Okay, next question is, an antibiotic resistance gene in a vector is usually help in the selection of what? Antibiotic resistance in the vector will be depend upon what, or it would be helpful in the selection of what? It is actually the transformation. We have seen the antibiotic. Which antibiotic we have seen? Ampicillin and the tetracycline and tetracycline, right? So these two we will use based upon the transformation, not the RDT or the competence sign. Okay, now next is the significance of the heat shock method in a bacterial transformation is to facilitate what? Heat shock means actually one cell was there, we will give the high temperature. Because of the high temperature there was the pores into the membrane, and the DNA or the material which was present into it, it will come outside with the pores, right? It was coming outside. That is what we have given the heat shock treatment. Okay, so heat shock treatment was actually to take up of the DNA through the transient pore in the bacteria cell wall. If we are doing this, or anything like any other bacteria cell, that also will have the pore, and the genetic material which has come out from here, it will enter into the, from the pores it will enter into the another cell, just like the transformation process which I showed you in the previous questions, right? So it is like that only. It is heat shock treatment, or calcium CaCl2 treatment also we was using, dicationic process. Okay, next question is 13. The role of the DNA ligase in the construction of recombinant DNA molecule. What is the DNA ligase function? Very simple question, because this is making a phosphodiester bond between the two DNA fragment. Absolutely correct, because it was actually making the bond between the base and sugar and the phosphate, right? So the sugar and the phosphate, this bond is known as the phosphodiester bond. It is actually the ester bond, phosphoester bond. Okay, and the bond between the base and the sugar is known as the glycosidic bond. This is the glycosidic bond. Okay, so why we are calling it phosphodiester? This is in the one strand, the same thing would be into the another strand, so that is why the two phosphoester bond, so phosphodiester bond. We'll go with the A. Then next question, which of the following bacteria is not a source of a restriction endonuclease? Restriction endonuclease का कौन सा source नहीं है? That is actually Ant amoeba coli. Ant amoeba histolytica was there, not the coli. Escherichia coli was there, Haemophilus is there, Bacillus amyloliquefaciens is there, but not the Ant amoeba coli. It is actually the combination of the two bacteria, so we cannot go with this one. Okay, which of the following steps are catalyzed by the Taq DNA polymerase in a PCR? Just now we was talking about the Taq polymerase in the previous question. So this was actually the at the primer ends, like we are having the two DNA strands which was attached with the help of the hydrogen bond with each other. This is the 5' end, this is 3', here is a 3', this would be the 5'. So we have given the high temperature, we have separated these two fragments from each other. Then we added the primer here in this part, and here also the primer, and then again we have started by using the Taq polymerase to extend the strand in the opposite ends, right? So on the basis of that, they are asking in the question, which of the following steps are catalyzed by the Taq polymerase? Taq polymerase are actually, this is the second step, no? This red one which I used, this was the step of the extension. This is known as the extension, right? So this is what they are asking into the question, that is the extension of the primer ends on the template by using the Taq polymerase. Okay, so we will go with the third one. Then next question is, a bacterial cell was transformed with the recombinant DNA molecule that was generated using a human gene. Means bacterial cell which is transformed with the recombinant DNA molecule and we are using a human gene. Okay, so the transformation of the cell did not produce any desirable protein. What could be the reason in that? Like bacterial cell is complete another genome, and human genome is completely different. Phylum is actually completely different. Monera and the we are having into the we are into the different phylum. They are into the Monera phylum. So how the genome can be matched first of all, right? So this cannot be possible, and the reason they are asking that human genome may have the introns. Yes, absolutely, we are having the introns and bacteria cannot have. This is correct. And amino acid codon for the human and bacteria, it is same, not the different. Like AUG will cause the methionine. AUG is a codon for the methionine, right? So it would be in the bacteria also, so it may not be the answer. Then human protein is formed but degraded by the bacteria. No, it is first of all not formed, then how can be degraded? So we'll go with the first one. Next question 17 is, which of the following should be chosen for the best yield if one were to produce a recombinant protein in a large amount? Then what we should use? We will use the continuous culture. The reason is, if we are having for example, this is the bioreactor, and in this if we are continuously adding the culture, new culture if we are adding, and from the another end we are removing the waste continuously. Adding culture is not a big deal, but the waste should also be removed with the time, because bacteria is growing, so definitely waste product would be there. So for that, waste product should be releasing. Continuous we are producing and then waste are removing, and then another time by particular time scale we will use the same procedure, and then the growth would be occurring continuously. So in that case, best yielding would be coming of the product by using the recombinant protein in the large amount. Large amount means by using the bioreactor, right? It is by using the bioreactors. So this process is known as the continuous culture. It is a continuous culture. One more is the another is process is the batch culture. In the batch culture, one time we are adding the media, and then after getting the product we will discard everything, and then again the next process we will start and continuing it. That would be the batch culture. Okay, next question is, who among the following was awarded the Nobel Prize for the development of the PCR technique? Herbert Boyer. Herbert Boyer was actually the discoverer of the RDT in the 1972, right? So they are asking for the PCR. Cannot be possible. Hargobind Khurana was actually, they have given the DNA sequence. It was given by the Hargobind Khurana, DNA sequence determined by him. Okay, or either basically we can say the amino acid sequence also. Carey Mullis has given the PCR, that is correct, and also the answer. Arthur Converge was actually discovered the DNA polymerase. Okay, DNA polymerase. Okay, so this is also not the answer. So we will go with the Carey Mullis. Next question is, which of the following statement does not hold true for the restriction enzyme? For the restriction enzyme, which is not true means which is incorrect, they are asking. Indirectly they are asking for the incorrect statement, right? So what could be the answer? It is isolated from the viruses, is it so? It is not isolated. Restriction enzyme is not isolated from the virus, from the Salmonella typhimurium. Okay, it was the Hind second was the first enzyme which was isolated from the Salmonella, which was a bacteria, not the virus. Okay, so it is incorrect, so we will go with this one. Okay, I hope these all questions up to here is will be clear to you, and let us meet into the next video with some other previous year question of the same chapter, and related to the next chapter of the biotechnology, second chapter of the biotechnology. Okay, I hope you all will enjoy with this. Thank you.